All right, back to decomposition. Sorry, there was a little bit of background noise there. So again, we're looking at how much variation is there within a species in terms of leaf litter decomposition. And from this figure, we can see that sometimes there's as much variation within one single species of plant as there can be across many genera and species within an entire plant family. And so this is an important source of variation for organic material. And let's see here. Here you can see organic matter, a timeline that shows major contributions to the understanding of organic matter processing in river systems from 1940. Of course, this is all Western ideas of how leaf litter decomposition occurs since kind of the development of the field of ecology. And so early on, we see things like Lindemann's work on trophic dynamics and Odom's work trying to measure energy flow in systems, some major leaf litter decomposition work. And what's exciting for me is that if you look up here at the top, we can see that that source of variation within a tree species made it onto this timeline of major contributions in terms of organic matter processing. So what might be some variation within species or across species? Things like these really large molecules like tannins and lignins, the different types of chemical bonds. So different species of tannins are made by different species and individual trees. How regular these structures are and repeatable. Some compounds are toxic. Have microbial toxicity, and so slow down those microbial colonization stages. Some compounds are sources of nutrients for bacteria and organisms on the surfaces of leaves. And, you know, in general, non-woody plant species tend to decompose faster than woody plant species on average. Things like nitrogen and phosphorus concentrations in leaf litter, which are nutrients that are resorbed by trees when they're losing their leaves. Those things can influence the speed of decomposition, tend to speed it up. Things like condensed tannins and other phenolic compounds tend to slow decomposition down, as well as structural compounds like lignins and celluloses. And then just how tough the leaf is, you know, is it evergreen, is it deciduous, is there a really thick waxy cuticle, is there not, how flimsy or sturdy the leaf is can all influence decomposition rates. And then this kind of the ratio between how much structure is there to how many nutrients are there, that can really determine decomposition rates as well. And so we tend to think stoichiometrically in terms of the carbon to nitrogen ratio of leaf litter, and the higher the C to N ratio, the slower the decomposition tends to be. So condensed tannins are really cool. They're, again, a complex recalcitrant molecule, not quite as big as lignin. They're a very common plant defense compound, and they can be induced by plants or by things that are attacking plants so that the plants produce more condensed tannins. They can inhibit digestion for both mammalian and insect herbivores, and they're also an antimicrobial agent. And so you can see already that they're probably going to influence decomposition rates. Tannins are potent antioxidants, so they keep things from being oxidized, right? And they're found in tea and wine, and again, this kind of staining of water. So tannins are one of the main causes for the brown staining of waters. The ecological influences of tannins, they can protect plants from herbivory. They may also deter shredders from leaf litter in streams. They can inhibit enzyme processes. They protect things like bark, heartwood, and leaves. And like I said, they have antibacterial properties, but they can also inhibit viral replication. There are studies of tannins inhibiting HIV replication. They can alter mycorrhizal associations between plants and their mycorrhizae. They can change nutrient cycling rates, and they can slow leaf litter decomposition rates. So lots of things. These are very biologically active compounds, really interesting. So here are two studies, or two figures from one paper showing that the decomposition rate is slowed for both aquatic and terrestrial decomposition. So as the condensed tannins increase, we see a slowed decomposition rate. We can also look at another study with a larger sample size showing decomposition rates slowed with increasing condensed tannins, increasing percent lignin, and no effect of the C to N ratio. So there's variation in these things. It's not always the case. Sometimes certain compounds will be stronger drivers in certain systems. Here you can see that we get the skeletonization of leaf litter. So what you're seeing is the loss of the labile components and the maintenance of those recalcitrant components, the veins, the structural parts of the leaf, the lignin remains for a lot longer than the waxy parts and the carbohydrates and all of the rich labile material. Interestingly though, if you look at a leaf litter, a piece of leaf litter through time, often the nitrogen content of the leaf litter increases. And that's not because the leaf material is more nutritious. I mean, it is, but the reason is that because microbes colonize the leaf surfaces and then they pull nutrients out of the water column for their growth and reproduction. And so because you can't scrape the microbes off the surface of the leaf before you measure the leaf chemistry, if you look at leaf chemistry through time, nitrogen and phosphorus tend to go up. And that's because of the microbes that have colonized the leaf surface. And so this is kind of counterintuitive thing. You'd think that nitrogen would just decrease through time, but it usually goes up through time. Again, because we can't separate the microbes and the fungal hyphal tissue from the leaf tissue itself when we analyze the leaf chemistry. I've said this already like 3 times, but lignin and cellulose are resistant to decay, and this results in this skeletonization. So the nutritional value of the leaf material that's remaining does eventually decrease through time. So once you get to the point where nothing's left but lignin and cellulose, it doesn't kind of matter how many microbes are on there. It's not going to be very nutritious for a shredding organism. Okay, so just a few studies. I've been really lucky. This is a terribly blurry picture. I apologize, but it's an old picture. I couldn't find a better one. I've been lucky to be a part of some really large collaborative global decomposition studies. So this is a whole bunch of us. We met in 2011 on 11/11/2011 to discuss this large decomposition study. We were pulling data from all kinds of other studies. So it was a big meta-analysis, and we were trying to understand the influence of global temperature changes on decomposition. And so this paper was led by Jen Folstead-Shaw, and it's this one here, this global synthesis of the temperature sensitivity of leaf litter breakdown in streams. But I've also been able to participate with some of the same individuals on this paper, forecasting the functional implications of global changes in riparian plant communities. And this paper, taking a plant phylogenetic history approach to explain decomposition at a global scale. And then another study, this was a totally different group of folks looking at mixed species effects, but another meta-analysis. And then this one with the most authors of any paper I've ever published. I think there are 157 authors on this paper looking at global patterns and drivers of ecosystem function in rivers and riparian zones using not leaf litter, but in this case, pieces of canvas, basically just cotton, which is mainly cellulose, to examine drivers of decomposition across hundreds of field sites. And so it was a really fun project to be a part of. And then just real briefly, I'll talk about a foundational study that really helped us understand how important leaf litter is to streams. So at the Coweta Long-Term Ecological Reserve Site, a research site in the Southeast in North Carolina, basically a leaf litter was excluded from one stream and compared to a control stream next door where leaf litter wasn't excluded. I don't know that this study, maybe it was started in 1993. That seems a little late, but I think that might be right. Anyway, what happened when they basically took all the leaves and woody material out of the stream and didn't allow any more to fall in. They created these barriers. What they found was that dissolved organic carbon in the water reduced by 30%. The stream insects all but disappeared. And yeah, I guess it was started in 1993. The first paper was published in 1997 by Wallace et al. And here's a picture showing the exclusion experiment. And what you can see if you look inside is that they tried to, eventually they started to replace the large woody debris, but with bits of things that weren't wood, just to see how, was it the structure or was it the actual like wood composition that was really important. And so you can see here, they put bits of PVC pipe of various sizes back into the stream after they had removed all of the organic material for quite a while. And then they slowly started bringing this woody material and leaf litter back into the system. So it's a pretty neat study to try to understand how streams are reliant on these allochthonous energy sources. So to kind of summarize the decomposition bit, decomposition is the major avenue of carbon loss from ecosystems. It's determined primarily by the same factors that regulate net primary productivity. It's very sensitive to global change, and there are potentially large feedbacks to climate change, things like methane releases from permafrost melting and from wetlands and that are warming. A lot of these connections to the global carbon cycle have to do with the fact with redox chemistry and with the oxidation of organic carbon. And so basically, if you start at the top, you have aerobic fungi and bacteria that oxidize organic carbon, and using oxygen, they turn it into carbon dioxide, right? But in the absence of oxygen, so especially if you go down in a lake or a wetland, then you have these other ways of oxidizing organic carbon, starting with nitrate and denitrifying bacteria that convert nitrate into N2 gas. And then when you run out of nitrate, then manganese reducing bacteria, then they become the most efficient. And then after you run out of manganese, iron reducing bacteria become the most efficient, and then sulfate reducing bacteria. And at the very bottom, you have the kind of fermentation byproducts and methanogenic bacteria that convert acetate and carbon dioxide into methane. And so you have this kind of like scale of efficiency in terms of the equation or the reaction moving through redox states. Okay, so to finish up with, methanotrophy is basically eating methane. So fermentation reactions in the absence of oxygen can yield methane and carbon monoxide, and methylotrophs will harvest energy by oxidizing these compounds, and they can keep methane from entering the atmosphere. So they're the ones eating methane. So that does happen to some degree, but then we also have methanogenesis. So other organisms can produce methane under anoxic and very low redox conditions. We think that ancient methanogens probably produce the natural gas deposits that we find deep in the earth. And then there are, in these ecosystems that are driven by methanogenesis, we have bacteria, methanogenic bacteria, that then get eaten by higher trophic orders. And in some lakes, we've found that Daphnia and other zooplankton can receive up to 50% of their carbon from methanogens. And we can follow these kind of trophic connections using stable isotopes. And then I just want to connect again to something I showed you earlier, which was these bubbles of methane in lakes that get frozen and trapped in the ice. And so this is one of the ways that we can measure how much methane is being released year round in these systems by looking at the ice and what's trapped when it's frozen. So just real fast, methane in systems can be sourced from natural sources, things like wetlands, lakes and rivers, wild animals, wildfires, termites, geological sources, but it can also be sourced from anthropogenic sources. So agriculture and waste, things like dairy farms produce a lot of methane, biomass burning, fossil fuels. And then where can this methane be stored? It can be stored in the soil, and it can be stored in the atmosphere, which is a place we don't really want it to be stored because then it acts as a greenhouse gas. So these are just kind of the fluxes that we've seen between 2000 and 2009. But global warming may increase fluxes, particularly from permafrost, from natural wetlands, and from streams as things warm up. Decomposition goes faster. And kind of putting it all together, here's an image from the IPCC report, the global carbon cycle. And what you can see are arrows going up are fluxes into the atmosphere, and arrows coming down are fluxes taking things out of the atmosphere. And this is basically carbon. So you can see the boxes then represent the source, like the pools, and the arrows represent the fluxes. And the red arrows represent human contributions to the carbon cycle. And so human contributions are majorly, the forms are fossil fuel burning, net land use change, and agriculture, and burning of forests for agriculture. So, all right. So it's really important that we understand the carbon cycle globally, and that we understand decomposition as a major part of this. You know, if we can figure out ways to slow down decomposition, we might be able to offset some of the changes to the carbon cycle. But we also, you know, have to recognize that decomposition is a natural process, and it's important for the functioning of a lot of aquatic ecosystems. All right. See you next time.